So basically, I graduated as an organic chemist and I became an enzymologist. So for almost like 10, 12 years, I studied classical enzymology. I worked on an enzyme. The name is calpain, so it's a really nice cytoplasmic enzyme. And this enzyme has an inhibitor that is fully disordered, but at the time, of course, we didn't have an idea about it. It was just, we always observed it's a different protein, it's a strange outlier. And then around 2000, and I had the idea, it was really nice to find the gene of this protein in the Drosophila genome. And that was the basic idea. When I started looking at the amino acid composition, the behavior, and it sort of struck me that this, strange protein is functional, but it doesn't have a well-defined structure. And I decided to work on this phenomenon, whatever it was. I didn't know that there was a field developing, or other people have already suggested some of it. And basically, I decided to work on this. And I started reading and writing, and I was sort of lucky because it turned out a very general phenomenon. So I was at that time in Budapest. So I'm Hungarian. I lived in Budapest most of my life, except for the last 3 years when I'm in Brussels, leading a department of structural biology. So this all happened in Hungary, and the idea came to me in Tokyo, actually. I was on a short sabbatical, and then I went home to Budapest, and I told it to my colleagues, that I sort of discovered something. It's very interesting, very intriguing. And they were the least excited by this. They said, I think it's not that important. Maybe it's just an artifact. You shouldn't be that, it's not a general phenomenon. And even if it's general, maybe it's not that important. So there was a lot of skepticism and a lot of, mistrust initially. So it took several years to convince them that it's becoming something very serious. And it's a very basic idea that sort of transforms molecular biology, structural biology around. Yeah, so that was it. So the nice thing about, doing bioinformatics or computational studies, that you are not limited to experimental things. You don't have to clone the protein. You're not really constrained to studying a single protein, but you can ask really general questions based on analyzing the sequence features, genome or proteome sequence features. And because of that, we could really ask many general questions, such as the role of protein disorder in chaperone function, the role of disorder in cancer-related fusion proteins, the role of disorder in eukaryotes, in prokaryotes, in heat adaptation, so many general things. So that's why we still like to do it. We keep continuing. Our latest, story that will come out is the analysis of degradation signals of proteins, which correlates with protein disorder a lot. So that's why we like to do a lot of bioinformatics, because it really gives you a lot of opportunity to ask general questions. One of the biggest challenges actually is to try to develop drugs against these disordered proteins, because it's pretty clear there's a very strong correlation between protein disorder and all sorts of, debilitating diseases like cancer, neurodegeneration, and some other stuff. So they are very attractive drug targets, as we say it. But the problem is that traditional means of drug development cannot be applied for these proteins. They don't have binding pockets, they don't have well-defined, binding regions. So somehow we have to target by small molecules this heterogeneous and very dynamic ensemble of these proteins. So it definitely takes new kind of vision, new approaches, new methods to do that. There are some successes already in the field, so people have developed some small molecule inhibitors of these disordered proteins. They are not very effective. In vitro they are effective, but in cell, in vivo setup, they are the least effective. So it doesn't seem to be there yet. So I think we have to apply new approaches. We have to sort of gather a new understanding of these proteins to do that. And we are working on such a project when a very important cancer-related drug target is disordered. It's a very important, cancer in human disease. If we could do something, that would be really a fantastic breakthrough. But it's a big challenge, as I said. It's very demanding. The basic point in this is that we have all grown up with something that we call the structure function paradigm, which states that the protein needs to be structured, to be functional. And if you want to understand the function of a protein, we need to be able to solve and understand the structure. and then connect features of the structure with certain elements or features of function. But this doesn't apply to disordered proteins. So another sort of recent challenge in the field is try to solve the structures of disordered proteins, which is a dynamic ensemble. So it's not a single structure, but a collection of 100 or 200 structures, and then try to understand function in terms of this ensemble. So connect structures of these IDPs with their function, and that's what we call unstructural biology. And the ambition of this new database is that we try to collect all relevant data. So we allow people to deposit their ensemble. Whoever solves an ensemble, they can deposit in the database. It's a fully open database, so people can have a look at these ensembles, they can analyze them, they can try to connect them with the functions, and they can try to improve, of course, our concepts of disorder function correlation. At the moment, what the database does, it just allows deposition of data, so it's a collection of data. But what we have to work on really hard now is to analyze this data. And neither, the points of analysis are clear, nor the tools of analysis are pretty clear. So we will have at this meeting, we will have a roundtable discussion, for example, about discussing what are the most pressing, questions in this regard, what would be the tools to analyze, and how to proceed with this whole thing.